Hello everyone, today, I will review a paper titled Microbial Fuel Cells, Methodology and Technology, written by Bruce Logan and his team. This paper is an essential resource for anyone studying microbial fuel cells, or MFCs. It provides a detailed overview of their design, materials, performance, and challenges while addressing gaps in research methodologies. By offering insights into standardizing evaluation methods, the paper aims to guide advancements in this promising field. Microbial fuel cells are an innovative technology that integrates energy production with environmental sustainability. They generate electricity while treating wastewater, making them a dual-purpose solution to two global challenges: energy scarcity and water pollution. Microbial fuel cells are devices that use microorganisms to convert the chemical energy stored in organic matter into electricity. Unlike conventional batteries, MFCs rely on living bacteria to drive the generation of power. Inside an MFC, microorganisms break down organic materials such as glucose, acetate, or wastewater. This process releases electrons and protons. The electrons are captured by the anode and travel through an external circuit to the cathode, producing electricity. Meanwhile, the protons pass through a membrane to the cathode, where they combine with oxygen to form water. This process is both an energy solution and a method for cleaning wastewater, which makes MFCs a highly promising technology for sustainable applications. MFCs can be used in wastewater treatment plants, remote power systems, and even marine sensors. However, MFC research faces the challenge of making comparisons across studies due to the lack of standardized evaluation methods. This paper aims to address these gaps by reviewing MFC design, materials, data analysis, and reporting methods to promote advancements in MFC research. Let's explore the various designs of microbial fuel cells discussed in this paper. The first design is the two-chamber H-type MFC. This setup includes two chambers, one for the anode and one for the cathode, separated by a cation exchange membrane. The membrane allows protons to pass through while preventing oxygen diffusion. This design is simple and widely used in laboratories for testing new materials and configurations. However, its high internal resistance often leads to low power density, making it unsuitable for practical applications. The second design is the air cathode MFC. In this system, the cathode is directly exposed to air instead of being submerged in a liquid catholyte. This eliminates the need for a separate cathode chamber and simplifies the overall structure. Air cathode MFCs are known for their ability to achieve higher current densities. However, they face challenges such as water evaporation through the cathode and oxygen leakage into the anode chamber, which can reduce efficiency. Lastly, there is the sediment MFC. This design leverages natural environments, such as marine sediments, to generate electricity. The anode is placed in organic-rich sediment, while the cathode is positioned in the overlying water. The redox gradient between the sediment and the water drives the generation of power. Sediment MFCs are often used to power low-energy devices, such as sensors, in remote or underwater environments. Each of these designs has its advantages and limitations, and the choice of design often depends on the specific application or research focus. The materials used in an MFC significantly influence its performance, cost, and long-term stability. For the anode, the key requirements are conductivity, compatibility with microbial biofilms, and chemical stability. Carbon-based materials, such as graphite, carbon cloth, and carbon fiber, are commonly used. These materials provide a large surface area for bacterial attachment, which enhances the efficiency of electron transfer. The cathode is responsible for reducing oxygen to water, a process that often requires a catalyst. Platinum is widely used due to its high catalytic activity. However, its high cost and limited long-term stability have driven research into alternative materials, such as manganese oxide, pyrolyzed carbon, and cobalt-based catalysts. These alternatives are more affordable and environmentally friendly. The membrane serves as a separator between the anode and cathode chambers, allowing protons to pass while preventing the mixing of oxygen and other ions. Nafion is the most commonly used membrane due to its excellent proton conductivity. However, its high cost has led researchers to explore alternatives like salt bridges and newly developed ion exchange membranes. Electricity generation in microbial fuel cells, or MFCs, is fundamentally a thermodynamic process. For electricity to be produced, the overall reaction must be thermodynamically favorable. This is measured using Gibbs free energy, which tells us the maximum amount of work that can be derived from a reaction. If the Gibbs free energy is negative, the reaction is favorable, meaning it can spontaneously generate energy. In the case of MFCs, the energy released during the oxidation of organic matter by bacteria is converted into electricity. This is quantified as the electromotive force, or EMF. The EMF represents the maximum theoretical voltage difference between the anode and the cathode. It is calculated based on the standard Gibbs free energy of the reaction and depends on factors like temperature, pressure, and the concentration of reactants and products. For practical purposes, the relationship between Gibbs free energy and the EMF can be expressed as a formula. It shows that the EMF is directly proportional to the number of electrons transferred in the reaction and the energy released. However, under real conditions, the voltage generated is always lower than the calculated EMF due to energy losses within the system. The EMF sets the upper limit for the voltage an MFC can achieve. By understanding thermodynamics, researchers can optimize conditions to maximize the energy conversion efficiency of the MFC. Now let's dive into standard electrode potentials, which help us understand the individual reactions occurring at the anode and cathode. In MFCs, the anode is where bacteria oxidize organic matter, releasing electrons. The cathode, on the other hand, is where those electrons are consumed, often by reducing oxygen into water. Each of these reactions has a standard electrode potential, a value that measures the tendency of a chemical species to gain or lose electrons under standard conditions. These potentials are reported relative to the normal hydrogen electrode, which is assigned a value of 0. For example, in an MFC using acetate as the substrate, the standard potential at the anode for acetate oxidation is negative, around -0.296 volts. This negative value indicates that electrons are released during the reaction. At the cathode, if oxygen is used as the electron acceptor, the standard potential is positive, about +0.805 volts under neutral pH. The difference between these potentials gives us the theoretical EMF of the cell, which can reach around 1.1 volts in ideal conditions. However, these potentials are affected by real-world conditions such as the pH and concentration of reactants. Understanding and controlling these variables allows us to optimize the electrode reactions, minimize energy losses, and improve the overall performance of the MFC. Voltage losses in MFCs are a critical factor that limits their efficiency and performance. These losses can be divided into three main categories: activation losses, ohmic losses, and concentration losses. The first is activation losses. These occur because energy is required to drive the redox reactions at the anode and cathode. At low current densities, activation losses are often the dominant form of loss. These losses are influenced by the choice of electrode material and the catalyst used. For example, using a more active catalyst, such as platinum, can reduce activation losses at the cathode, though this comes at a higher cost. The second is ohmic losses, which arise from resistance in the various components of the MFC. This includes resistance within the electrodes, the membrane, and the electrolyte. Ohmic losses are proportional to the distance between the anode and cathode, so reducing this spacing can help minimize these losses. Additionally, using materials with higher conductivity for the electrodes and membranes can also improve performance. The third is concentration losses, which are caused by mass transport limitations. These occur when the supply of substrate or oxygen to the reaction sites cannot keep up with the demand, leading to a drop in reaction rates. For example, if bacteria at the anode deplete the organic substrate faster than it can diffuse through the system, the rate of electron generation will decrease. Similarly, at the cathode, insufficient oxygen supply can slow the reduction reaction. Improved reactor designs, such as enhanced mixing or flow systems, can help mitigate these losses. Each of these losses impacts the overall voltage and power output of the MFC. By understanding the sources of these losses, researchers can develop strategies to reduce them and improve system efficiency. To optimize MFC performance, researchers rely on a variety of measurement techniques to analyze the system's electrochemical processes. These techniques provide insights into how the system operates and where improvements can be made. The simplest and most common tool is the multimeter, which measures the cell voltage. While this gives a basic overview of performance, it does not provide information on internal dynamics, such as resistance or electron transfer efficiency. For a more detailed analysis, researchers use a potentiostat. This device allows for advanced techniques such as cyclic voltammetry, current interrupt tests, and electrochemical impedance spectroscopy. Cyclic voltammetry is used to study the electron transfer mechanisms and the activity of the microbial community or catalyst. In this test, the potential of the electrode is swept through a range, and the resulting current is measured. The shape of the resulting curve, or voltammogram, provides information about the redox reactions occurring in the system and the effectiveness of the materials. Current interrupt tests are used to measure ohmic resistance in the MFC. By momentarily interrupting the current flow, researchers can analyze the resulting voltage drop. Immediate changes in voltage reflect ohmic losses, while slower changes correspond to activation losses. Electrochemical impedance spectroscopy is a more advanced technique that evaluates the dynamic properties of the system. By applying a small alternating current and measuring the system's response, researchers can identify resistance, capacitance, and other factors affecting performance. These tools and techniques are critical for diagnosing problems, optimizing designs, and improving the overall efficiency of microbial fuel cells. The performance of MFCs is evaluated using several key metrics that balance energy production with environmental impact. One key metric for evaluating MFC performance is power density, which is calculated as power divided by electrode area or reactor volume. Using voltammetry data, we can also generate polarization and power curves. This graph is a polarization curves and shows the relationship between voltage and current density and help analyze major loss factors in MFCs. Polarization curves are typically divided into three regions. This graph is a polarization curve, typically divided into three regions. First is the activation loss-dominated region, where voltage drops steeply as current begins to flow. Second is the ohmic loss-dominated region, where voltage decreases more gradually in a linear fashion. Third is the concentration loss-dominated region, where current rapidly decreases. The graph below is a power curve, which illustrates the relationship between power density and current density. Peaking at the maximum power point. As the current increases, the power also rises until it reaches the maximum power point. Beyond this point, power begins to decrease due to increasing ohmic losses and electrode overpotentials. Besides, there are various metrics for evaluating the performance of MFCs. The first metric is coulombic efficiency. This measures how effectively the electrons released by the substrate are captured and converted into current. Coulombic efficiency is calculated as the ratio of actual current output to the theoretical maximum based on the substrate's oxidation. A high coulombic efficiency indicates that the system is effectively utilizing the electrons generated by the bacteria. The second metric is treatment efficiency. This evaluates the system's ability to remove organic pollutants, typically measured as the reduction in chemical oxygen demand, or COD. High treatment efficiency is critical for wastewater applications, where the primary goal is to clean the water while generating energy. The third metric is energy efficiency, which is the ratio of the electrical energy output to the chemical energy of the substrate. This metric provides an overall assessment of the system's ability to convert organic matter into usable electricity. By analyzing these metrics, researchers can assess the trade-offs between energy production, environmental impact, and cost-effectiveness. Microorganisms in MFCs transfer electrons to the anode through three main mechanisms: direct transfer, shuttle-mediated transfer, and nanowire-based transfer. The first mechanism is direct electron transfer. Certain bacteria, such as Geobacter and Shawinella species, use specialized proteins on their cell membranes to transfer electrons directly to the anode. This method requires physical contact between the bacteria and the electrode and is highly efficient because it eliminates the need for intermediates. The second mechanism involves electron shuttles, which are small molecules that carry electrons from the bacteria to the anode. These shuttles can be naturally produced by the microorganisms, such as flavins and quinones, or they can be externally added, like methylene blue or neutral red. This method is advantageous because it allows for electron transfer even when bacteria are not in direct contact with the electrode. However, it can introduce additional costs if external mediators are required. The third mechanism is nanowire-based transfer. Some bacteria, like Geobacter sulforeducens, produce conductive appendages called nanowires. These structures act like tiny electrical wires, transferring electrons from bacterial cells to the anode or even between cells. Nanowire-based transfer is particularly interesting because it enables the formation of dense microbial communities, enhancing overall electron transfer rates. Understanding these mechanisms is critical for improving the design of electrodes and optimizing microbial activity. By enhancing electron transfer, researchers can increase the efficiency and power output of MFCs. In conclusion, microbial fuel cells represent a groundbreaking technology that combines renewable energy production with environmental sustainability. They offer the potential to address two pressing global challenges: energy scarcity and wastewater pollution. However, significant challenges remain. These include improving power output, reducing material costs, and scaling up the technology for real-world applications. Future research should focus on developing cost-effective materials, enhancing biofilm performance, and optimizing system designs to minimize losses and maximize efficiency. As advancements continue, MFCs could find applications in diverse fields, from wastewater treatment and biosensors to portable energy systems and marine devices. With ongoing innovation, MFCs have the potential to play a key role in building a more sustainable future. That concludes my review of the paper Microbial Fuel Cells, Methodology and Technology. Thank you for your attention, and I hope this presentation has provided valuable insights into this fascinating and promising technology.